Thank you for standing by and welcome to today's Chimeric Therapeutics webinar, discussing yesterday's announcement of a new Phase 1b clinical trial for CHM 0201 in newly diagnosed AML. All participants are in a listen-only mode. There will be an initial presentation lasting approximately 20 to 25 minutes, followed by a Q&A. If you have a question that you'd like to submit, please feel free to type it in using the Q&A function within the Zoom app. On behalf of Chimeric Therapeutics today, we have the CEO and Managing Director, Jennifer Chow, Chief Medical Officer, Dr. Jason Litten, and the Principal Investigator of the trial, Dr. Abhishek Maiti. To begin, I'll hand it over to Jen. Please go ahead.
Thanks, Matt. And thank you, everybody, for taking the time this morning to join us as we talk about the ADVENT-AML trial that we announced yesterday. We're incredibly excited about this trial, and so we're thrilled that you've joined us to learn a little bit more about it. Just before I do that and hand it over to Dr. Maiti, I want to just actually give a little bit of intro to the trial, how it came about, and just talk very briefly about where the company is today. So for most of you, you're well aware that our company has three novel platform technologies. The first, CHM 1101, is our chlorotoxin CAR-T that's being studied in recurrent glioblastoma. And we're very much looking forward to presenting the Phase 1a preliminary data later this fall. CHM 2101 is our CDH17 CAR-T, and this is a first-in-class CAR-T that actually is targeting gastrointestinal tumors, and we are in the process of moving that towards clinic. But we're here tonight to talk about CHM 0201, which is our allogeneic NK platform, and the new clinical trial that is going to start shortly using that platform. Now, with all of our technologies, we are very aware of the share price sensitivity and certainly the pressure that we see on our share price today. There's a lot of challenges in the biotech market generally. You see highlighted here just some of the statistics that have come out recently. There's been an incredible decrease in the biotech IPOs that actually are moving forward. About 20% of the current biotech companies that are listed on NASDAQ are actually trading for less than the cash that they have on hand. And only 29% of companies in both the US and in Europe have cash reserves to last a year. So more than 70% of companies don't. So that's the biotech market that we're obviously operating on. And certainly we unfortunately have not been able to escape the challenges of that market. So I just wanted to highlight a couple of things briefly that we're doing to really make sure that we are doing everything we can at Chimeric to be able to take off some of the pressure that we're seeing on our share price currently. First and foremost, we've done a lot of work about program prioritization. With the three novel technology platforms we have, there is a lot of opportunity for development in multiple different types of cancer and combinations and next generation technologies. And our team has worked really hard to make sure that we are focused in the right areas. The second is we're very cognizant of cash preservation. And so we've really been looking at keeping a very small, lean team. We do not have any office space, and we're very, very responsible when we think about our fiscal situation. And then the third is that we've really been exploring business development opportunities. And some of these are collaborative opportunities. And some of these are opportunities like what you've seen recently in our ability to find technologies and actually pass them over to other companies, bringing in the license, the finder's fee from Imugene. So this is some of what we're doing to make sure that we are doing our best as a company. The other thing that we know with the biotech market today is that the bar has gone up for the expectations for biotech companies. And really what I mean by that is nowadays, people really want to see clinical data. That preclinical stage data is just no longer enough. And so when you think about and highlighted here, the stages of clinical development, sort of moving from preclinical in vitro into in vivo development, into Phase 1a, which is very much focused on safety and dose escalation, then Phase 1b, Phase 2, and ultimately Phase 3, we know that the bar has moved and very much people at least want to see Phase 1a or Phase 1b data. And certainly, historically, often preclinical data was considered enough. For us, we're actually very much focused on making sure that all of our programs are moving forward from a clinical perspective. And certainly Jason has spent an incredible amount of time and energy ensuring that our programs are streamlined and optimized clinically. From our 3 novel technologies, all of them now have completed all of the preclinical work, both in vitro and in vivo. 1101, our chlorotoxin CAR T, has completed now its phase 1A dose escalation and moved into phase 1B. And CHM 0201, the NK cell platform we're going to talk more about this evening, has also completed its phase 1A clinical trial and now moved on to combination trials as phase 1B trials. CHM 2101, our CDH17 CAR T, we anticipate moving into clinic next year. And so you'll see as we talk about our milestones and the events that we have coming forward, very much our focus is on making sure that we're advancing our clinical programs to actually meet the higher biotech bar nowadays. So with that said, now what I want to do is really spend all of the rest of the time that we have this morning talking about our NK program. And just for everybody's reminder, our NK cell is an allogeneic NK cell platform. And by allogeneic, what I mean is that it's really off the shelf or on-demand therapy. So essentially what we use is we use a healthy donor, and the healthy donor actually gives us blood. We isolate the NK cells, we can engineer those NK cells, and then we actually freeze them so that they're available on demand as a patient would need them. And so this is an allogeneic or off-the-shelf platform. The platform that we at Chimeric have, CHM 0201, we believe is a highly differentiated NK cell platform. And you see some of the science highlighted here and why we believe it is differentiated. It uses a unique genetically manipulated feeder cell line as the basis for expansion, and we already have an FDA master cell bank. We've been able to demonstrate robust expansion of our cells and potential for up to 1,000 doses. And we've already seen through the existing phase 1A data, activation, expression, and persistence of these cells. So we really do believe that this particularly NK cell platform is one that is really differentiated and something we can build off. I'll just remind you of the phase 1A clinical trial that was completed at Case Western, and this has already been published. Within that trial, they actually looked at patients who had both acute myeloid leukemia and colorectal cancer. And what we saw first and foremost come out of that trial was safety. We really wanted to be able to look at when you take cells from one healthy donor and you actually go and you give them to another patient without matching them, can you do that safely and without what we call GVHD or graft versus host disease? So this study was very important in that it established that could be done with this platform. We saw persistence, and persistence really means that the cells are staying alive so that they can do their job inside a patient. So importantly, that was an endpoint for this trial, and we saw 28-day persistence of the cells. And we also saw expansion. So we were able to manufacture number of doses from one single donor. From an efficacy perspective, in the colorectal cancer patients, we saw some disease control. About 1/3 of the patients were able to achieve disease control. And with the blood cancer patients, all of the patients were able to achieve disease control. But keep in mind, these are very small numbers of patients because this was a phase 1A trial. We were also very pleased to see with one of the acute myeloid leukemia patients what we call a complete response. And that patient we know now has been able to sustain that complete response for more than 2 years. So for us, this was an incredibly strong foundation to build off. And we really then wanted to spend time thinking about what is the right direction and how do we really build off this. We focused our efforts in 3 specific areas. One was to take the platform that had been used in the phase 1A trial and look at, could we put it in combination with other therapies that actually may be able to work synergistically and enhance the efficacy that we were seeing in the phase 1A trial. The second way that we're looking to develop the 0201 platform is actually by engineering it. And so we're looking at CHM 0301 as our next generation platform, where we're doing some enhancements to it so we can actually have a next generation armored platform. And then what we're working on is actually taking our chlortoxin and our CDH17 CAR, engineering them into the 0301 platform to develop CAR NK products in the longer term. So that's really how we've built our plan. Now, the combinations, first and foremost, you probably remember in January of this year, we announced the first combination trial using CHM 0201. And that is a trial that is going on currently at Case Western University in Ohio. It's enrolling patients that have colorectal cancer and acute myeloid leukemia. And what it's doing is it's putting CHM 0201 in combination with an agent called bactisertib. And so we're looking forward next year to seeing how patients in this trial are doing. What we're here this evening to talk about, and I'm going to introduce our principal investigator in a moment, is the next combination trial, what's now known as the ADVENT-AML trial. And I'll let Dr. Maiti talk to you a little bit about how he came up with the trial and the name. But I just wanted to give a little bit of history. So when we actually brought CHM 0201 into Chimeric, Dr. Maiti actually contacted us and asked us whether or not we would be interested in putting our NK cells in combination in AML patients, as he believed that there was really good, strong scientific rationale that perhaps can improve the efficacy that he's seeing in his practice. We were thrilled when he actually reached out to us, and we certainly have been working hard with him, and Jason and Abby have been working very closely together over the course of the last 9 months or so to develop this trial to where it is today. But we were thrilled because Dr. Maiti has been incredible to work with. And so we thank you very much for that, Abhi, as well. And certainly you see highlighted here just some of the incredible achievements that he's had in his very young career. So his focus very much is a clinical and a research focus on developing new therapies for patients that have leukemia. He's already authored several peer-reviewed publications in leading medical journals and has presented multiple times at national and international scientific meetings. He's received recognition from ASCO, or the American Society of Clinical Oncology, from EHA, the European Hematology Society, from ASH, the American Hematology Society, and many others. And then you see highlighted just a few of the awards that Dr. Maiti has also already been able to achieve. So we were very excited to be able to work with someone that has had such an impact already in the area of leukemia and novel therapies for patients with leukemia. With that, what I'm going to do now is I'm actually going to turn this over to Dr. Maiti to be able to take you through a little bit of background on the trial, the rationale for the trial, and his thoughts about how he came up with the design of the trial and what he's planning to do with it in the future. So Abhi, thank you so much for joining us this morning. We very much appreciate your time, and I will turn it over to you and look very much forward to hearing from you about the trial.
Thank you, Dr. Chow, Dr. Litten, for inviting me and for the generous introduction. I'm very glad to be here with all of you today and talk about this very exciting study. So Jen, if you could move forward the slide to the trial, wonderful. So this trial, it's a combination of current standard of care with azacitidine and venetoclax with the core NK cells. And the name is based from the alphabets in azacitidine, venetoclax, and NK cells. And it also sort of symbolizes the tremendous advancement that this trial, I believe, will contribute to this field. So on the, I will keep some of the discussion very high level, but please feel free to ask me any detailed questions. So on the left side, what you can see is basically a vast majority of patients with acute myeloid leukemia need transplant. Transplant basically reboots their bone marrow and provides them an immune system for the rest of their life, which can fight the leukemia. However, acute myeloid leukemia is also a disease of older and unfit patients. The median age is 68 years. At most centers, I would say majority of patients are unable to proceed to transplant because in these older patients who are often very frail, weak, they have other medical problems, transplant can cause more harm than benefit. Anywhere from 1 in 5 to 1 in 20 people die because of the complications from transplant. However, and here you can see, and this is the first data set we published, and then there was another data set from the International VLEA trial group where they showed that this majority of patients who receive azacitidine venetoclax, they're not candidates for transplant. So 80 to 90% of patients are ineligible to proceed. However, our center is very aggressive, and with these very small numbers, we showed that patients who are able to proceed to transplant have significant benefit. The amount of time they can live, it's more than doubles. And the risk of death decreases by 50%. So what got me thinking and why I reached out to Jen is that how can we get patients this benefit without getting them through the rigorous and toxic process of transplant? So that's where I came up with the concept of combining the current standard first-line therapy with allogeneic NK cells. And that's what the figure shows you on the right side. Jen, if you could move to the next slide. So here, and then, once when I was developing this concept over a year back, and everything came together so beautifully in the sense that this trial, the data, the preclinical and clinical data that this trial is based on, that spans decades of work by leukemia physicians and scientists, and also very cutting-edge novel data that was published last year, which sort of was the final data point, which made me feel that this is so compelling that we have to do this trial. So to go briefly over the rationale, all these immune therapies, at least in leukemia space, they usually, and even in lymphoma space where these are approved, but they're not approved in acute myeloid leukemia, these usually work when the disease burden is very low. But when our patients come to us, they usually have very high disease burden, their bone marrows are packed with leukemia, and these immune cells don't have a chance to work. So combining these cells with the standard of care azacitidine and venetoclax provides a compelling rationale that the standard treatment will bring down the disease burden, and then the NK cells, the immune cells, will have a better chance of working. And then the other rationale is that these medicines, the azacitidine, what that does, it upregulates proteins on the surface of leukemia cells, making those leukemia cells more detectable to immune cells, and thereby making them susceptible to immune-mediated killing. And with this treatment, what we are doing on the left middle side, you can see that we are triggering multiple arms of cell killing. So there are recognized arms where the cell is killed by an intrinsic mechanism, excuse me, which is called BCL2, which is targeted by venetoclax, this pill, which was developed in 2017. And the azacitidine also works to kill the cells internally, while the NK cells will help to kill the cells from outside, so the extrinsic pathway of cell killing. And then there was the recent published data, which was published in part from our group and then from Dana-Farber group in Boston, which showed that venetoclax synergizes with NK cells. So venetoclax sort of primes the leukemia cells for killing. And that was, when I read that paper, I felt that, this trial had to be done. And then, I was fortunate to have Jen and Jason sort of understand that rationale. And then the final idea, which I mentioned, is that when patients benefit from transplant, the major benefit is from what we call graft versus leukemia, that the new immune system continues to fight leukemia cells. So my thought was that it would be incredible if we can deliver that immune effect only without other risks and bypassing all the morbidity and mortality risk from allotransplant. So all these rationale came together to form the background for this trial. Next slide, Jen. And this is some preclinical research. On the left side is data which showed that how venetoclax, this oral pill, synergizes with immune cells, NK cells to be specific. And on the middle slide, you can see that in patients, in mice with acute myeloid leukemia, these mice live longer. And on the right panel, it shows that we had to confirm that our cells are resistant to venetoclax so that we can combine them. So Dr. David Bald, who manufactured these cells, his laboratory confirmed that our cells are truly resistant to venetoclax, which enables us to combine this in this triplet therapy. Next slide, Jen. And here is the other piece of very compelling data, and a lot of this was generated at MD Anderson. So our investigators in the lymphoma group were the first to sort of lead the approval of CAR T cell therapies in lymphoma. And what was so fascinating was initially CAR Ts, these immune cells were tested in patients where multiple lines of treatment had not worked, and then they were slowly brought up to the front line. So as you can see that when these immune cell therapies, the CAR T cells therapies, were brought from third or fourth line therapy to frontline therapy, the response rates jumped over 20%, and patients who were essentially cured of this disease at 1 year jumped by 30%. So this tells us that a newly diagnosed hematologic malignancy is more susceptible to eradication by immune cells than a disease which has been relapsed or refractory after prior therapies. And that's a major challenge in this field, because when all these companies are going after leukemias and lymphomas with these immune cell therapies, they are all focusing on the relapsed refractory population where the disease is so difficult and so biologically so difficult to make a dent that these treatments are not working. So that's where we come in, that if we can get this treatment into frontline, which we plan to with this trial, I think we're going to see significant benefit compared to what we have been seeing with trying these therapies in the salvage setting. Next slide, Jen. So this is a brief summary of the trial. So because this combination is so novel, it's sort of high risk, high reward, we have to start out in the relapse refractory setting where the potential benefits far outweigh the potential risks. We plan to treat just 6 patients to demonstrate to the US FDA that this is safe. And then we're going to have, and I personally feel that this will be safe based on everything we know about these cells, the mechanism of synergy. And once we have that data, then we'll quickly move this to frontline adverse risk AML patients, where we think we'll be able to show a benefit in a short time frame. And just to give you a background, excuse me, acute myeloid leukemia, it's an orphan disease. It's a rare disease. It's considered an orphan disease by governmental agencies because it's rare. It's also an area of high unmet need. And the bar of drug approval is very, very low. So to give you an idea that venetoclax was the biggest improvement in the field, and we sort of led the development of venetoclax in AML. So our group has led the development of, I think, around 9 out of the 10 last FDA approvals in AML space. So we certainly have a lot of experience in this, but despite all those improvements, we're not curing patients. So if you look at the adverse and intermediate risk group of AML, who comprise more than, I would say, 60 to 80% of patients, the 4-year overall survival is still less than 10-15%, which tells us that all these advancements, they are only enabling patients to live a little bit longer. But what we are truly trying to do with this very novel combination is we're truly trying to cure these patients. I believe that what we will see a significant jump in remission rates, which we'll find out very early. Within a span of a month, 2 months, we will have that early signal. And then by 1 year, following patients for 1 year, at that time, we'll also get the other early signal of how long these patients are living, because even at 1 year in this adverse risk population, the amount of patients who are alive, it drops down to 30-40%. If we can show that a significant proportion of our patients are living more at the 1 year time point, that will truly tell us. And that is what Jason asked me when I pitched him this trial, that you can do this trial, but how are we going to know that we're making a difference, that we're improving outcomes? The way we will do it is that we have very contemporary controls, and we can do rigorous matching, so sort of a retrospective randomized trial, and demonstrate the benefit that we are hypothesizing this triplet will give us. And so my job is until then to demonstrate that clinical proof of concept, and then I'll sort of hand it over to the Chimeric team so that we can plan for a pivotal trial and think about an FDA approval. And going back to that same topic, that AML being an orphan disease, being area of high unmet need, there is potential that if we can show, I think if we can show early benefit, we can go to FDA and ask for a breakthrough therapy designation. And if we can enroll more patients and continue to see that benefit, even with a 80 to 100 patient trial in this rare disease, FDA has historically given out accelerated approval. And that's my goal. My goal is not simply to do this trial, but how can I get this therapy to patients across US and the world? So that's in a nutshell the trial design. And Jen, if you move to the next slide. These are additional very sophisticated single cell analysis that we will perform to truly understand how this treatment is working, because the beautiful thing about NK cells is they just don't go in and kill leukemia, but they also teach the host immune cells how to kill leukemia. So we'll be able to demonstrate that with this very sophisticated testing that we're planning to do on these samples. Next slide, Jen. And here are some of the nitty-gritties about the clinical trial design. Happy to answer more questions about exactly, what we will measure and how we will demonstrate that this treatment is improving upon the current standard of care. Next slide. And this is the incredible team. I'm so grateful to be working with from Chimeric Therapeutics, Case Western, and a tremendous group of investigators at our cancer center, and funding from Gateway, Chimeric, and our institution, which will help to get this trial going. So that's in a nutshell. I'm happy to take any questions.
Yeah, thank you so much, Dr. Maiti. That was a really impressive presentation, and this is a really exciting study and an important opportunity to help potentially AML patients who, as you described, really are in need of new treatments. I have a couple questions for you, and then maybe we can address some questions from the audience as they come in. I was hoping you could maybe describe for us, you mentioned that patients with AML who aren't eligible for transplant are generally older. I was wondering, can you just provide a little bit more detail and description on the types of patients or the typical patient you might expect to enroll in this clinical trial? You mentioned that they're on the older side. Are they very ill? Are they ambulatory? Are they sitting in your waiting room, or are they hospitalized? Can you give us a sense? And maybe there's some variability there that you can describe as well.
Thank you, Dr. Litten. Great question. So, like I mentioned, that AML is a disease of elderly patients. The median age is 68 years. So you can imagine 50% patients are older than that. And the way AML usually presents is these patients are incredibly sick and it develops rapidly. These patients were doing fine one time, and then within a few weeks, days, weeks to a couple of months, these patients are bedbound. They're severely anemic, they're having shortness of breath, and sometimes they get severe infections and they frequently end up in the ICU. Us being a very large referral center, our department here is literally accepting patients from across the state and all the neighboring states where patients are getting transferred from hospitals and ICU. So direct hospital to hospital transfer, ICU transfers. So these patients usually present to the emergency room because they're feeling so sick. I would say there's a minority of population, usually among the younger patients, who, for miraculous reasons, get diagnosed early, maybe incidentally. But those patients are few and far between. Vast majority of patients are incredibly sick when they present.
And that's actually very helpful. Thank you. Maybe actually spending a couple minutes reflecting on MD Anderson in particular. It's a very special place, doing some of the most important cancer research that's going on right now. So I understand that a lot of your patients, and you already kind of mentioned this, aren't local to Houston. Can you describe, again, the patients that you expect to be treated in this clinical trial? Will they have traveled for the trial? Are they here with, are they in Houston with you for an extended period of time? Are they with caregivers, and do they stay in the hospital for the whole time they're with you, or do they have alternative accommodations?
Great question, Dr. Litten. So yes, so we are the largest leukemia program in the country and probably the world. So we get referrals for newly diagnosed and relapsed refractory patients from, of course, the state and all the neighboring states and all around the country, even from, from some of the other premier cancer centers. When they have exhausted options, they travel here. Houston is a large city, so we have a substantial proportion of patients who live in Houston, but a large number of acute myeloid leukemia patients do end up traveling from outside. And that's being because MD Anderson is known for our AML program. So vast majority of patients, particularly the newly diagnosed patients, they actually spend the first month in the hospital, because the first month is where a lot of things can go wrong. And outside MD Anderson, sometimes the risk of death in the first month is anywhere from 10 to 30%. So outside large academic centers, there's a sense of nihilism, and these patients don't even get treatment because the risk of death and poor outcomes, and they are sent to hospice. But here, that risk of death in the first month is less than 5% because we monitor our patients so closely in the hospital. And then after that period, vast majority of our patients were treated on clinical trials. They have a lot of resources and housing within Houston, which are housing specifically for patients getting treatment at MD Anderson. So vast majority of patients stay in and around Houston while getting treatment.
Great, thank you. And that's actually a good segue to spend a couple minutes reflecting on these patients' current treatment options. Do they have choices? Are there options you're able to present to them, or is it far more limited? And when you have these conversations with these patients, are they expecting a cure, or are they hoping to just extend their life by a few months? And I imagine there's variability there too.
Great question, Dr. Litten. So if you take into account all patients with acute leukemia, particularly our population, older unfit population, roughly 20% of them have one targetable mutation called FLT3. Another 15-20% have targetable mutations called IDH. And the rest do not have anything actionable. Vast majority of places outside large academic centers, they have been treating leukemia with the same chemotherapy for the last 40 years, and that hasn't changed. It's only at large academic centers for those 35 to 40% patients, we're able to offer them a little bit different approach, combining targeted therapies, but vast majority of them are not standard of care, and those are on clinical trials. For FLT3 mutant patients, we reported the first data on the triplets. And then for IDH mutant patients, our group led the development of IDH inhibitors. So that's one chunk. And then for the vast majority of the remaining patients, 60 to 70% patients, there's no targeted therapy. So the standard across the country and the world is azacitidine and venetoclax. But something like this has a tremendous opportunity to improve upon that.
That's great. So maybe we can talk about that for just a minute. What does improvement look like? You mentioned the endpoints that you're going to be evaluating in this study, but what do those endpoints mean to patients? Are we talking about an improvement in their life, an extension of their life, and why would a patient be excited about this study?
Great question, Dr. Litten. So the reason I became a leukemia doctor is because when I used to see patients with AML, I used to get scared because these patients are so sick. But the beautiful thing about our field is that AML is curable. This is so different from all other solid tumors, lung cancer, breast cancer, any other solid tumors where if the disease has progressed, it is not curable and patients will unfortunately die from their disease. But acute myeloid leukemia is a curable leukemia. So that's our goal with this treatment. Currently in the adverse risk group where we're going to first go into with this treatment, we see remissions, so responses in anywhere from 45 to 55, 60% patients, but up to 80% of those patients relapse. So what we're looking to see with this treatment is by that 1 month, 2 month endpoint, we're looking to bump up that remission rate by at least 15, 20% and bump up really deep remission, which we call MRD or measurable residual disease, which is using a much more powerful test to truly identify, if there's any traces of leukemia left. We're looking to increase that deep remission rate by, again, 15 to 20% over 40, 50%. And we were the first to show that deep remission correlates with significantly longer survival. And anytime we're talking about survival, these survival figures include patients who pass away from a car crash, so that not to introduce bias into those estimates. But my goal with this treatment is to move that bar where the long-term cure rates are less than 10, 15%, to modestly improve them by at least 20%, to go to 35%, which would be a massive, massive development for the field. But if this works as well as we hope it will, based on the compelling preclinical synergy, the cure rates may be higher.
That's great. Thank you for that. And maybe just one more question about the clinical trial. And I know that I and the rest of the audience is very results oriented. How long do you think it's going to take to accrue the study?
A great question, Dr. Litten. So we see the highest volume of patients. So the number of patients we're looking to enroll on this clinical proof of concept is 6 patients relapse refractory and 20 patients newly diagnosed. We see 40 of those patients every month. So, we can theoretically enroll all the patients in a month, but because this is such a novel area and no one has done this before, no one knows the safety, that's why FDA requested us to stagger patients. That is, the first 3 patients, we'll need to enroll them and wait for a month to make sure this is safe. So the first 3 patients will take 3 months. The 4th patient will take another month, and the last 2 patients in that 6th patient will be enrolled in the next month. So the first 6 patients, we will, by FDA mandate, we will need 5 to 6 months to enroll. And then again, we want to select the patients properly because the entire AML community, the leukemia community, will be looking at this trial. So we want to select those patients properly. So 6 months for the dose escalation phase, and for the 20 patients dose expansion, we want, again, because we want to select the patients properly, although we can enroll that number of patients in a month, I think it would be reasonable to say that because we're also going to open this at Case Western where they don't see as much volume, I want to say at least over a period of anywhere from 4 to 8 months, conservatively speaking, we want to slowly enroll those patients. We also have to wait for cell manufacturing and other things.
Great, thank you. We do have one question from the audience. The question is about the incidence of AML in the US annually. And I won't hold you to the 2022 statistics, but a rough estimate. And then maybe if you could provide your conjecture as to whether or not this treatment, and I'm assuming the questioner is reflecting the combination of the 3 drugs, has the potential to treat other indications.
Great question, Mr. Hovey. So the annual incidence of AML in the US is around 20,000, and it's very slowly increasing every year. We don't know why. So but that still makes it an orphan disease. And whether this treatment has the potential to treat other cancers, great question. So, we think this will be a practice-changing trial for the field, not only for AML, but all hematological malignancies and solid tumors, by the virtue of bringing cell therapies in the frontline setting, leveraging this beautiful synergy. So the other disease, which I can think of right off the top of my head, where this specific combination can help is T-ALL, T-cell acute lymphoblastic leukemia. In adults, there are around 2,000 cases a year, but it's more common disease in pediatric population. And it's also an area of high unmet need, patients with relapsed refractory disease. On an average, they survive for 4 months. So that's an area where I would love to explore this specific triplet. And then venetoclax plus NK cell therapy, so the 2 diseases out of 2 drugs out of these 3, that has very broad potential because venetoclax is applicable to so many different diseases. So we joke that we love venetoclax so much that if we would add it to the Houston water supply, but I don't think the authorities will allow us, because so many cancers depend on the protein that venetoclax blocks. And there's an incredible number of trials going on in different hematological malignancies, and there's rationale for solid tumors. So this combination can be translated to multiple different cancers. We can start out with some of the lower hanging fruits and then move to other diseases. And the other very close cousin of AML is myelodysplastic syndrome, which is sort of a pre-leukemia, but within that, there are high-risk subtypes. So that's another very low-hanging fruit because NK cells are of interest in there. AZA-VEN trials are currently being run in that disease setting, and that would be a very nice disease setting to test this triplet. So MDS, T-ALL, some other orphan diseases, biphenotypic leukemias, and then any of the other trials that where chimeric platform is currently being evaluated, including a neuroblastoma and others. The combination with venetoclax can definitely be entertained.
I think that's a great point and a very complete answer. Jen, did you have any questions you wanted to pose to Dr. Maiti? Okay. And Dr. Maiti, do you have any closing comments?
Yeah, I genuinely appreciate the help from the Chimeric team and all the people behind Chimeric who have helped us with the funding for this and the manufacturing of the cells, without which we wouldn't have been able to do the trial. And I cannot tell you how much grateful our patients are for having access to this so novel treatment approach.
That's great. Well, you're very welcome. It's a pleasure to work with you. And thank you for your time and for your really important work taking care of cancer patients. So thank you. Jen, if you'll maybe just go to the last slide where I can just reflect on what you talked about at the outset, which is our pipeline, which is very active. You mentioned our program CHM 1101, which is continuing to enroll patients with advanced brain tumors. CHM 2101, which is our autologous CAR T program that's going to be going into the clinic next year. And now we have an updated pipeline slide, which is in blue, our NK cell therapies that are either in the clinic or going into the clinic. We now have a new line for the AML study at MD Anderson. So we'll probably update that and continue to follow Dr. Maiti and his team's progress at MD Anderson very closely. Thank you for your time. And with that, I think we can close the call.
All right. Thanks very much.
Thank you, everyone.
Thanks, Jason. Thanks, Dr. Maiti, and to you too, Jen. As you've mentioned, that concludes today's Chimeric Therapeutics webinar. And thanks to everyone else in the audience for joining today. And we'll look forward to bringing you more soon. But goodbye for now.
Take care, everyone.